Good evening, everyone. Good evening, and welcome to yet another super cool and super amazing last session from the chapter Mineral Nutrition. And today's class, lecture number 4, we are going to learn about the steps in nitrogen fixation and about the fate of ammonia. Okay, ready, everybody? Those who have not become a part of our Telegram channel, don't forget to click the link below and become a part of our Telegram channel. So let's get started with the very first topic of the day, that is about the nitrogen fixers. Okay, so what are the nitrogen fixers? Nitrogen fixers can be about the bacteria, it can be about the archaebacteria, or it can be the cyanobacteria. So nitrogen fixers or the nitrogen fixing organisms, it can be simply bacteria, or it can be organisms belonging to archaebacteria, or it can be cyanobacterian groups. So if it is coming under the bacterian group, then you can call them as the rhizobium, frankia, acetobacter, mycobacterium, azospirillum, and bacillus. If it is coming under the archaea group, then methanococcal, methanobacteriales, methanomicrobiales, cyanobacteria, nostoc, anabena, etc. These are not mentioned. Okay, so what is mentioned in the NCERT, I'll highlight that for you so that you can study those things. That is about the rhizobium, it have been mentioned, about the frankia, it have been mentioned, acetobacter, azospirillum, bacillus, then about the anabena and nostoc. That is about the cyanobacteria, and about the normal bacteria have been mentioned as nitrogen fixers in our NCERT. So just study the terms since we have detail learned in the last class about what is rhizobium, what is frankia dis, what is acetobacter and azospirillum dis. So we are going to discuss about the steps that have been involved in the process of nitrogen fixation. The first process is the ammonification. In the last class, we have studied about what is nitrogen fixation, the conversion of atmospheric nitrogen into ammonia, right? The conversion of atmospheric nitrogen into ammonia is known as nitrogen fixation. Now, what is ammonification? What is ammonification? Decomposition of organic nitrogen of dead plants and animals into ammonia. Conversion of atmospheric nitrogen, that is the organic nitrogen of dead plants and animals into ammonia, you will call them as ammonification. Okay, whereas some of them re-enters the atmosphere itself. Okay, so guys, the plants and animals by the action of the soil bacteria. So conversion of the dead plants and animals into soil bacteria into soil is known as decomposition. From the soil bacteria, it will undergo the process of ammonification. Finally, ammonium ion or ammonia is formed. So the process by which ammonia is formed from dead and decaying plants and animals are known as ammonification. Next step is the nitrogen fixation. Next comes the ammonification. Next comes the nitrification. What is nitrification? Nitrification means, that is, we have already learned that by the process of ammonification, ammonium ion have been formed, right? By the process, you have learned that ammonification, ammonium ion is formed. So ammonium ion is converted into nitrite, NO2 minus, nitrite ion by bacterium such as Nitrosomonas or Nitrococcus bacteria. Very, very important, Nitrosomonas or Nitrococcus bacteria. Nitrite is converted into nitrate with the help of the bacteria Nitrobacter. So 3 steps are involved. Ammonium is converted into nitrite. Nitrite is converted into nitrate. Ammonium into nitrite facilitated by Nitrosomonas or Nitrococcus bacteria. Nitrite into nitrate is facilitated by the Nitrobacter bacteria. Clear? Very important. You must study the importance. Bacteria name, very, very important. Okay. After nitrification, that is now what is a product in a hand? Nitrite and nitrate. So next step is the denitrification. That is a conversion of this nitrites and nitrates formed back into atmosphere as atmospheric nitrogen. So that process is facilitated by 2 different bacteria, that is Pseudomonas and Theobacillus. You must study what are the bacteria that helps in the process of nitrogen fixation. We studied what is the bacteria that helps in the formation of ammonia to nitrite, nitrite into nitrate. We have studied Nitrosomonas, Nitrococcus, and Nitrobacter. What is the name of the bacteria that converts nitrites and nitrates into nitrogen? Denitrification process also we have studied right now, that is Pseudomonas and Theobacillus are the two bacteria which helps in the process. This will be a short session because there is nothing in detail which I have to keep on repeating. The things over here are just for you to get it by heart. Is it done, everyone? Name of the enzymes. Nitrate is converted into nitrite by the enzyme nitrate reductase. Nitrite is converted into ammonia by nitrite reductase. So nitrate reductase and nitrite reductase are the enzymes. Clear, not mentioned in the NCERT, you just have to understand over the same. So ammonia, and we have to study about the amino acids over here as well. So there are two ways by which the amino acids are synthesized. One is the reductive amination and second one is the transamination. What are the two methods? Reductive amination and second one is the transamination. Clear? First, we have to discuss about the reductive amination. Okay, what is the method known as reductive amination? Okay. Reductive amination. What is reductive amination? The ammonia which has been formed react with a product or react with a compound that is alpha ketoglutaric acid. Alpha ketoglutaric acid, very important, alpha ketoglutaric acid, and forms glutamic acid Okay, very important, that is the ammonia. Okay, the ammonia which have been, one second, let me change the color of the pen so that it can be clearly visible for you. Okay, the ammonia which have been formed will react with the product that is the alpha ketoglutaric acid. Alpha ketoglutaric acid and activated by the enzyme glutamate, dehydrogenase, activated by the enzyme converting itself into a compound called glutamate. Very important, you have to study 3 to 4 things over here. One more thing, one more time I'm repeating. Process reductive amination, ammonia reactant react with alpha ketoglutaric acid, forms glutamic acid. So alpha ketoglutaric acid plus ammonia in the presence or mediated by the enzyme glutamate dehydrogenase. Very important. What is it? Repeat it along with me on the chat box. Glutamate dehydrogenase converting into the product that is the glutamate. Done? So that is how ammonia is converted into an amino acid. Next method is a transamination. That is, it involves a transfer of the amino group from one amino acid to the keto group of the keto acid. Okay, glutamic acid is the main amino acid from which the transfer of NH2 group, the amino group, takes place, and other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reaction. Is that clear? So this is converted to this, this is converted to this by the process of transamination. And which is the enzyme that catalyzes the process? It is transaminase. Is this clear? Is this idea clear for everyone? Is it clear? So one more time, let's discuss or quickly recap what all things that we have learned in today's class. So firstly, we have discussed about the ammonification process, that is how decomposition of the organic nitrogen of the dead plants and animals is converted into the process of ammonia. And next one is nitrification. Nitrification is the process by which ammonium is converted in nitrite into nitrate. Okay. Okay. and about the different types of bacteria also we have studied in today's class, about the reductive amination and about the transamination. Finally, we have a small portion. I have cut out the same from the NCERT, the two important amides, that is asparagine and glutamine, found in the plants as a structural part of the protein. Okay, they are formed from two amino acids, namely the aspartic acid and the glutamic acid respectively, by the adding of the amino group by the method of transamination. By the method of transamination. Clear? So, since amides contain more nitrogen than the amino acid, they are transported into other parts of the plant via the xylem vessels. In addition, along with the transpiration stream, the nodules of some plants, such as a soya bean, export the fixed nitrogen as urides. Okay? So that's in this chapter, mineral nutrition. And in the very next day, we will be starting with the next chapter of plant physiology, that is photosynthesis in higher plants. Bye-bye, guys. See you. Thank you so much.